Welcome to 1.2. In this unit, we're going to talk about the macromolecules. What macromolecules make up cells and their organelles. We're going to focus on carbohydrates, lipids, proteins, and nucleic acids. This unit is going to be broken up into 3 segments because there's a ton of information I got to give you. Let's get on with the first set, carbohydrates.
So last time we talked about carbon and the functional groups associated with carbon, and now we're going to talk about how those carbon backbones are able to form macromolecules. And we're going to look at some of the properties of all macromolecules. And it does start with that carbon backbone. Add to this, we have some functional groups which give macromolecules certain properties that they may have. And along with these functional groups and carbon backbone, macromolecules are made up of repeating units known as monomers. These monomers combine by dehydration synthesis to form polymers. Long complex chains of repeating units. These polymers can then be broken back down into monomers by hydrolysis. We're going to look at this in more detail. There are 4 classes of macromolecules known as lipids, proteins, carbohydrates, and nucleic acids. We're going to look at each group in more detail, starting with the carbohydrates. But first, let's look at dehydration synthesis, how we build up monomers into polymers. With dehydration synthesis, otherwise known as condensation reactions, 2 monomers are joined together by the removal of a water molecule. This water is pulled off as a hydroxyl group and a hydrogen ion, and those 2 combine to form water. This leads the 2 monomers open to covalent bond with each other, forming a bond by dehydration synthesis. For all organisms, this process requires energy and enzymes. More about energy in a later unit. Hydrolysis, on the other hand, also requires enzymes. However, energy is released during this process. It is the opposite of dehydration synthesis. We are going to add water and split it into a hydroxyl group and a hydrogen ion and break that bond and put those 2 pieces back onto the monomers, as noted where the blue arrow is. Hydrolysis is the process of digestion. When you digest food, you're breaking substances down into small enough pieces so that your cells can absorb them. In your cells, they're going to build up monomers by dehydration synthesis when you build up carbohydrates for storage or proteins to do certain cellular functions.
I have a little trick to help you remember the difference between hydrolysis and dehydration synthesis. Little trick here, get your fingers, they're your monomers, okay? Everybody needs to get their fingers up, even you. Don't roll your eyes at me, okay? These are your monomers. For dehydration synthesis, it goes like this. Remove a water, form a bond. For hydrolysis, add a water, break a bond, okay? Say that with me one more time. Remove a water, form a bond. Add a water, break a bond. Okay? That little trick should help you remember the difference between dehydration synthesis and hydrolysis.
So the first group of macromolecules that we're going to look at are carbohydrates. The monomers of carbohydrates are known as monosaccharides. These are simple sugars that can be made up of 3, 5, or even 6 carbons. And please note that your 6 carbons have several different structural isomers that you can have. And this is going to lead to some biological differences and how your body is going to digest them and how they are going to be formed into polymers. All sugar names are going to end in OSE. That's how you can tell something is a carbohydrate, if it ends in the suffix -ose. In cells, because it is an aqueous solution, a water-based environment, the monomers are not going to be in a linear form. They are going to be in a ring structure, as shown here. The alpha and beta forms are two different isomers of glucose. This plays a key role in how they are joined together in the polymer. There's the alpha, and there's the beta structure. Notice just one little minor difference in the structure. Two monomers are going to come together by dehydration synthesis to form what we call a glycosidic linkage. A glycosidic bond or glycosidic linkage is the bond formed between two monosaccharides. Glycosidic linkage is just a fancy name for the bond formed by dehydration synthesis in a carbohydrate. Sucrose is formed between a glucose and fructose monomer. It is commonly known as table sugar. Other disaccharides that are important to organisms are maltose and lactose. Maltose is going to be two glucose molecules joined together by that glycosidic linkage, and lactose is glucose and galactose joined by that glycosidic linkage. Notice the difference in structure. With lactose, individuals who cannot digest dairy because of the sugar in it cannot digest lactose. They do not have that enzyme. And so lactose is broken down by various bacteria in the intestinal system, which leads to some unpleasant symptoms. Whenever you have multiple monosaccharides joined together in long chains, we form polysaccharides. One of the most simple is starch. And starch is primarily made up of alpha glucose isomers. And the key function is energy storage, and they are found in plant tissues. Amylose and amylopectin are two forms of starch. They are all alpha-based, and I want you to look at the bonds in red. These are your glycosidic linkages, and notice they all go in the same direction. Organisms have no trouble breaking these bonds. We, in fact, as animals, store glucose in the form of glycogen, just like amylose and amylopectin, except it's highly branched. It is still made up of alpha glucose monomers, or alpha glucose isomers. The key role is to store extra energy in the liver and muscles of animals. On the other hand, you have structural polysaccharides. The key role of these are to give support to cells and tissues. Cellulose and chitin are two forms of structural polysaccharides. These are non-living structures, and they are formed by the beta isomer of glucose and the beta glycosidic linkage. Chitin is found in the exoskeleton of arthropods, insects, crustaceans, organisms that have that hard exoskeleton, and it's also found in cell walls of fungi. The key difference is that there's a nitrogen group coming off one of the side chains of the glucose rings. Cellulose, on the other hand, is just beta isomers of glucose. And I want you to notice that every other glucose is upside down in order to form that glycosidic linkage. Animals do not have the enzymes to break this down for energy. Certain organisms have developed symbiotic relationships with bacteria and protists so that they can extract the energy found in the carbon-carbon bonds of cellulose. For us, humans, we do not have the symbiotic bacteria. However, cellulose is an important for proper functioning of our digestive tract. This is what we call insoluble fiber. Its key role is to keep our digestive system functioning and to keep everything moving through properly. This concludes the introduction to monomers and carbohydrates. We're going to continue on in another video with lipids and nucleic acids.
All right, you just got done with part 1 of 1.2. What you should have learned was that monomers form polymers by dehydration synthesis, and polymers can be broken down to monomers by hydrolysis. The other thing you should have learned were the types of carbohydrates, their monomers, and their polymers. Which ones are used for energy storage and which ones are used for structural purposes. You got 2 more parts to go, so keep on going.